Welcome, everyone. Thank you for joining us for today's track on Earth and human management of our microbial wealth. It's my pleasure to introduce to you Dr. Jonathan Scheiman. Jonathan is a co-founder and CEO of FitBiomics, a biotechnology startup that is sequencing the microbiome of elite athletes to identify and isolate next-generation probiotics for applications in consumer health and nutrition. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Scheiman's keynote presentation is titled Mining Athlete Microbiomes for Next Generation Probiotics. My name is Hillary Thompson, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots. Labroots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Jonathan Scheiman. I will now turn the presentation over to him.
This is Jonathan Scheiman, co-founder and CEO of FitBiomics. It's a pleasure to be here on Labroots and have an opportunity to talk about our research that's leading to sort of our company, FitBiomics. High level, we're a biotechnology microbiome early stage startup. We spun out of the Harvard Medical School a little over a year ago. And basically, at a high level, what we do is we sort of look to sort of mine the microbiome of elite athletes to sort of identify next generation probiotics that are either unique or enriched in them, and see how those probiotics could potentially, drive optimal performance and recovery, but then ultimately sort of use this metagenomic information and translate it into real world consumer products to help promote health to the masses. So, I'll sort of go through today, basically my background story, why I got excited in sort of microbiome research and why athletics, but also talk about some of the science that was recently published in Nature Medicine, and then talk about future applications and, where we could go from here. So I guess with that, we could get started. I always, kind of like to start at the beginning. I think every story has a beginning. I mean, I'm a scientist, I'm an entrepreneur, but I wasn't always that. I actually was a basketball player. I always tell people, first and foremost, I'm a New Yorker. I was born and raised in New York City. And actually, I played basketball at St. John's University. So this is a Division I sort of a basketball school. Well, it's a school that does a lot of things, but I think was also known for its basketball program. I was a walk-on. We had pretty good teams. I was there from 99 to 03. We won a Big East championship. We won an NIT championship. And for me personally, I think growing up in New York with hoop dreams in the city, I wanted to play in the NBA and be a professional basketball player. There's some interesting statistics associated with this, and I think this has been covered, on the internet and online when I tell my story. But, not a lot of people get a chance to play in the NBA, so I didn't make it, but my backup plan was getting a PhD in molecular biology. So that's how I became a scientist, sort of as a, because my primary options didn't really become available. But as a scientist, and this is kind of a bit of a joke, I am a big believer in, natural selection and form fits function. And, I look at these statistics and I think it's easy to sort of laugh at these things now. But even from a scientific perspective, if you look at sort of people that are able to overcome these odds, now a lot goes into this, but if you think about this from sort of a physiological perspective, these are basically, super athletes are extremely beneficial phenotypes. So, what if we were able to understand what separates like, Stephen Curry from myself, other than, talent and training and greatness, but maybe from a biological perspective, are there any differences that could, enable certain athletes to overcome these odds? But then, of course, beyond just athletics, what implications does this information have for maybe just, overall human health, performance, and recovery? I think society in general, and I like to sort of play around with this, we're heavily influenced by, athletes. I think obviously this is a play on certain commercial from the 80s and 90s. But I think in general, athletes have a profound sort of impact on our daily lives, not just in terms of sports or being fans or, watching TV, but even in terms of sort of emulating their performances, how active we are, exercise, certainly even in terms of like economic decisions, things that we eat, things that we drink, things that we buy to wear. So, it is interesting, the impact athletes have on our daily lives. And, the notion of how do we actually be like these athletes? How do we emulate them? I think right now the current paradigm is, we have to eat what they eat. We have to drink what they drink. We have to wear what they wear. I think one question is, does any of this really get us closer to not only emulating them in terms of their peak, performances, but really, do these things get any closer to optimize our own personal sort of performances? And, I think obviously coming out of, the Harvard Medical School and the Wyss Institute for Biologically Inspired Engineering, I think we take a little bit of a different approach with FitBiomics and the research we're doing is, what can we learn from an elite athlete's, biology? And again, how can we sort of use that information to maybe drive, consumer health and nutrition? I think there was a joke I like to tell, like early on, I think, I spoke with an NBA general manager, and when I was first telling him what we do from a biotech and genomics perspective, I think the first question he asked was, can we use genomics to predict the next Michael Jordan? And, I think my initial and almost immediate response was no. But, maybe to be a little bit more provocative, I think the question I asked him was, well, can we sort of understand Michael Jordan's biology, and maybe sort of extract that and give it to other people to help maybe develop the next Michael Jordan, or maybe even just develop, health, solutions or options for people, even outside of athletics. So, the notion of like working with the most fit and healthy people in the world, understanding them, and then translating that for everyone else. I'll take a little bit of skipped ahead. I'll take a little bit of a detour here. Talking about understanding optimal physiology, optimal phenotypes, optimal biology, and decoding that information. I think for me personally, this is the real reason why I became a scientist, the notion of genotype to phenotype, the notion of form fits function. And I think for me, I was always excited about the diversity of biology in terms of different organisms, different animals, different species, and how each has somewhat evolved to sort of have unique, physiology that may be relevant for their environment, that may be relevant for survival. And I think what really excited me early on is sort of like understanding that from a molecular basis. And I think, when you get into sort of the genomic era, and I guess we're still, maybe, 15 years or 16 years since the first human genome sequence was published, there's obviously still a lot of information out there that we could learn from. But this is a concept that's really, I think, influenced me as a scientist and sort of something that excites me a lot, understanding the blueprints from a genomic standpoint for different, life forms. And, I will say this, I was a basketball player, I'm a scientist, I'm very big into movies, I'm very big into pop culture. And, this is, something, the notion of everything around us, life, it could be architecture, it could be sort of governments, various forms of society, but everything sort of having a structure, everything having a blueprint, everything having sort of a code. And, really what's exciting is, again, what are the implications and the potential applications of understanding those codes? I mean, imagine, being able to sort of see the underlying framework from everything around you, and then being able to sort of interface with that to optimize, human health or things of that nature. And obviously, this is a scene from, The Matrix, which I love, and I'm excited that they just announced a Matrix 4 that's coming out fairly soon. But, just even from a health perspective and understanding genetic blueprints, genetic codes, molecular foundations for human health and disease, imagine understanding all that and then being able to sort of engineer it or edit it to sort of promote health. And, I love this line, what are the applications of understanding the matrix? Or, I guess we'll use this from, molecular foundation for life. Can we dodge bullets? But no, if we could understand these things and implications for health and disease, maybe we won't even have to dodge bullets anymore. We could sort of engineer optimal health, and that could be through different modalities. And I think really that's what's the heart of biotechnology and biomedicine right now. Again, full disclaimer and disclosure, I am really into pop culture. Here's a fun fact. In addition to sort of being a basketball player and a scientist, I grew up in Comic-Con industry. Pretty much every year of my life, I've gone out to San Diego Comic-Con, and I've seen that community develop, from just like a niche cult following that's strictly sort of comic books to now what it is today, where it's all Hollywood and all movies and, billion-dollar franchises, and Disney owns everything. But, to kind of take this a step further from blueprints for encoding life, I think a lot of times, certainly in the biomedical community, we're very interested in understanding disease. My PhD was in molecular oncology, and, can we understand disease? What doesn't work, and can we correct it to promote health? But what if we took an opposite approach? What if we looked at, for all intents and purposes, super beneficial phenotypes or superheroes, right? And what makes these, superheroes unique and perform novel tasks? Can we understand that from a molecular basis, from a genomic basis? And can we learn from that? I'm huge fans. I mean, this was just in the last year, Black Panther, Captain Marvel, Spider-Verse. I'll just admit I saw Endgame in theaters probably 4 times. I've probably already seen it around 6 times. So heavily influenced by the genre, but I think that's also exciting because, I actually did a postdoc because I was heavily influenced by Iron Man and Tony Stark, and I saw the lifestyle of him and basically using technology as a superpower. And I thought, wow, that would be a pretty cool lifestyle. So I'm a big believer in, this will tie back into sports, but just sort of popular culture being infused into science and technology and creating sort of new functional categories that could be relevant to the masses. We like to think of these superheroes as make-believe, but we'll tie it back into athletics, right? Are superheroes real? And going back to the point, 0.00001% of people that play in the NBA, and it's not even just the NBA, it could be other sports, but for all intents and purposes, these elite athletes are superheroes, right? I mean, they run faster than us, they jump higher, they're stronger, their mental toughness is off the charts. Again, the foundation for understanding beneficial phenotypes and physiology and optimal health, I think, nature is already selected for these phenotypes. And now is there a way to work with them to sort of understand from a biological perspective what actually works? And I think this is, the underlying foundation for FitBiomics and sort of the research that we're doing. Can we, sort of work with the most fit and healthy people in the world? Can we understand what makes them unique and perform at an optimal level? Can we then sort of mine that biological information to develop, consumer products that can promote health to the masses? And, it's a fairly straightforward concept, right? Work with athletes, sort of use next generation sequencing to decode their biology and then translate that. I will, again, a major theme in this and sort of state this again, I'm a former athlete, so there's a lot of meaning for me to work with the athletic community. I think there's also a lot of, cool collaborations that can happen between science and pop culture and athletics as a medium. But just even from a scientific perspective, working with elite athletes is a proxy for understanding optimal health. And I think that's something that we're excited about. And also just say, we're a microbiome company. So I'm a little biased, but I happen to believe that the microbiome is one of the more translatable areas of research right now. It's something that is dynamic. It's something we could change with diet. It's something that we could extract, something that we could transfer. It's something that we could purify. And, if we're talking about sort of beneficial bacteria, that's just basically another word for probiotics. So really for us, we want to mine the microbiome of elite athletes to look for next generation probiotics that we could then purify, functionally validate, and to look to turn into food, beverages, and supplements that everyone could sort of consume to help make them more fit or active or just support their daily lifestyles. I think everyone sort of knows this by now. I don't know if I need to spend too much time, but it's kind of a provocative notion that we're more bacteria than human. This number keeps changing. I think it used to be we were 10 times more bacteria. We were 2 times more bacteria. Now it may be we have as much bacterial cells as human cells. And I'm not sure where that final number stands, but I do know that we have trillions of bacteria in and on our body. And I do know that this ecosystem is collectively known as our microbiome. What I'll say is that, this is a continuously growing field. And, for FitBiomics, we're pretty much focused on gut bacteria. Certainly, there are more microorganisms in the gut, fungi, yeast, viruses. And certainly, if you look at a microbiome, what's also exciting is it extends beyond, sort of the gut. There's obviously oral microbiome, skin microbiome, nasal microbiome, hair microbiome, vaginal microbiome. So I think this whole community of microorganisms, is just continuously expanding in terms of, what we're discovering. Again, I think from FitBiomics' perspective, again, coming out of biomedicine and my background, I think the paradigm in biomedicine right now is look at disease physiology. What doesn't work? What's broken? Can we correct it? Again, I think our approach is almost the exact opposite. Let's look at what does work and extremely healthy and beneficial phenotypes, certainly from their microbial community. And what are the applications? Over the past 10 years, I think this has been firmly established that our microbiome greatly influences our health, our development, our functionality, certainly from an early age all the way up to adulthood and beyond. I think as a scientist, we look at these from a perspective of energy metabolism, protein metabolism, neurology, immunology. I think if you look at from an athlete's perspective, we're talking about endurance, strength, mental toughness, and recovery. So again, really what FitBiomics is sort of doing is, again, let's work with sort of phenotypes that nature has selected for being optimal performers. Let's try to look to identify beneficial bacteria or probiotics that could drive these physiological processes. Can we see what's unique or enriched in athletes, and then sort of, again, look to translate those into real world probiotics? I mentioned I was really in George's lab at the Harvard Medical School for 6 years. I initially did some in situ sequencing and CRISPR-Cas9 research, but then I decided to go back to my roots in athletics, and then pretty much for around 4 years worked on sort of athlete microbiome technology. And we spent a lot of time recruiting, marathon runners, ultra marathon runners, rowers trained for the Olympics. We sequenced their microbiomes. We see how they compared to non-athletes. We see how they compared from to athletes from different sports. But what's really exciting, as this cartoon depicts, is we actually track these athletes in a longitudinal manner to see how their personal microbiome changes between performance and recovery phases. And then ultimately, we look to sort of mine all that metagenomic information and then look for, again, bacteria that are either unique or enriched or, change over time. And, sort of look into this information to see, okay, this is interesting. This is exciting. Why don't we see if we could purify these bacteria and do some functional testing? What's really exciting is, this this culminated in a Nature Medicine paper that was recently published, I think around 2 months ago, where we discovered some, and this was done in George's lab. It was at the Wyss Institute for Biologically Inspired Engineering. It was also in collaboration with Alex Kostik's lab at the Joslin Institute for Diabetes. This was really exciting. And, I'm going to now talk about this research, obviously starting with, lofty goals and, talking about athletics and comic books, but, now getting to the science aspect of it, where we discovered basically a bacteria whose natural function is to eat and break down lactic acid, convert that into metabolite that's beneficial for the host, and then can actually promote endurance and recovery in preclinical settings. So it's kind of crazy, it's kind of cool, but I think this is sort of the nature of science. Like I said, this all started around 4 years ago in George's lab at the Wyss. And, I just remember the initial conversations. How do we design a project around this? How do we start it? This was actually called a sports genomics project, and it was IRB approved, and we spent time designing it. It actually started back in 2015 when we recruited athletes from the Boston Marathon. And I should also just say, too, what goes into sort of designing this study, obviously there's sort of like selection criteria, inclusion, exclusion criteria, what determines if someone, is an athlete, how many times a week do they work out? Also, it involved recruiting sedentary controls, which, honestly, we're mostly just, scientists around the community. But, enrolling them into this IRB study, the other thing I should say, too, is, obviously, if we're looking at the microbiome, the gut microbiome, we're collecting samples that passage through the GI tract. So this is just a fancy way of saying we collect, hundreds of fecal samples from athletes. I think a fun fact is 60% of fecal material is around bacteria. So literally in each sample you collect, there's millions of microorganisms in there that provide a wealth of information. And we can sort of, decode that. But we do more than just collect fecal samples. We collected a ton of metadata. We collected hundreds of answers on a food frequency questionnaire about, their dietary habits, their health background, their athletic background. And then every day we collected a sample. We had a daily annotation sheet where participants, told us what they had for breakfast, lunch, dinner, any snacks, their exercise routines, how many hours of sleeps they had, at what time they collected their samples. So there was a ton of information that we collected for this study. This is somewhat convoluted, but I'll just high-level background for sort of microbiome analysis. And what we do is, I mentioned, we collect stool samples. The great thing about biotechnology right now is how sort of accessible a lot of products are to facilitate research, especially in this space. So you could buy a commercially available kit that its function is to basically extract genetic material from feces. So we could collect that genetic material, and this is predominantly from the bacteria that's in stool. Once we have that genetic information, we could sort of modify it in a format where it's suitable for next generation sequencing. Basically it's sort of taking raw material and then giving us sort of genetic sequences, A, T, C, G. And once we have all that information, then we sort of go to, use bioinformatic techniques where we could run it through sort of search engines and reference databases, almost like, we're running sort of genetic sequences through like a Google, if you will. And then what that does is then it sort of spits back out all the organisms that these genetic sequences are derived from. So you can imagine you collect one stool sample from an athlete, and now you have a list of all the microorganisms that are in their gut. Now you can see how that changes over time, and now you can compare that to other athletes. You compare that to non-athletes. And again, you have all this sort of information. And at a very high level, this is kind of, what it looks like. I remember, again, this graph is maybe from 4 years ago. And this is from the marathon study. We had several hundred fecal samples that we had to process, and sort of get back genetic information. I think this initial data set was using a publicly available software called Scheiman, readily used by the American Gut and, Rob Knight's lab, that they developed this software. And basically each color here, well, first of all, each column represents a different fecal sample that was collected, from an athlete. And then basically, each color represents a different type of bacteria. And I think when you first look at this information, it's very messy and it's hard to know basically what to make of this. But this is why you need to work with talented sort of bioinformaticians and statisticians, because they could sort of look for signal in all of this noise. And as I mentioned, this was a project that I collaborated with Alex Kostik with. I actually met him when he was a postdoc at the Wyss Institute, and then sort of how he transitioned into being a professor. And again, I remember I sort of provided him the raw data, I think around summer of 2015. And then shortly thereafter, he came back to me with some initial slides to show what he found from all this data. And this was actually one of the first graphs that he showed me. And basically, what we did was we recruited around 20 marathon runners. We recruited around 20 sedentary controls. We collected fecal samples on a daily basis for 2 weeks, before the marathon, after the marathon. I should also just say something about Boston. For whatever reason, it's enriched in endurance athletes, whether that's running or rowing. And I think people in the Boston area know that the Boston Marathon is a huge, huge deal. It's a holiday. It's on Patriots Day. My first day working in George Church's lab happened to be on, the marathon Patriots Day. Actually, I think my last day in his lab was around that time. And it's only, fitting that I think halfway in between, that was the foundation for this paper. But anyhow, we collected these samples. Here's another fun story, just as on a tangent, while we're talking and while we're being honest and open and having fun here. There's a lot of practicalities to consider when running these studies. And there's a lot of logistical sort of situations that you don't necessarily think of when you're sitting in the lab. So, we collected fecal samples on a daily basis, for, I don't know, 20 to 40 participants. What does that mean? That basically means for 2 weeks, I was driving around Boston in a zip car, 5 to 7 hours a day collecting fecal samples. So, I like to say I got my hands dirty for this project. But you got to sort of be willing to sacrifice some things for the sake of science. But anyhow, I'm sure you've already read this and you've seen this slide, but one of the interesting things that popped up with this is when we looked at sort of these runners, you could see here the blue line are our runners, right in the middle is the day of the marathon. One thing that sort of popped out was we found right after the marathon there was a spike of this bacteria. But the interesting thing was its natural function was to eat and break down lactic acid. I think this was a really big light bulb moment for us because lactic acid, although the literature, I think, a lot of people, I think lactic acid, people still aren't sure what it does, or at least there's some misconception about what it does in terms of causing fatigue or burning sensation. And actually, it turns out it's an energy source and is mostly a byproduct of exercise. But in general, lactic acid is a metabolite that accumulates in the blood after intense exercise and is associated with fatigue, not necessarily something that causes it. But basically, here we found almost a biological response in the microbiome to exercise, whereby, right after an intense workout or the marathon, there was an increase in a bacteria that sort of eats a byproduct of exercise that accumulates in the blood. So this was kind of exciting, but it kind of had our heads thinking for a little bit, well, what are the potential applications for this? What if we had a bacteria that we could purify as a probiotic? And what if we could give it to athletes before they ran a race and could potentially, prevent fatigue, promote endurance or promoting recovery? So again, I could just say after spending hours upon hours upon hours upon hours of collecting fecal samples, getting this sort of result was somewhat rewarding and validated and made the whole thing worthwhile. But it kind of gets better from there. That sort of analysis was done with 16S, ribosomal DNA, very high level, looks at a genus. We then took it a level further. We did comprehensive metagenomic sort of shotgun analysis of these fecal samples. And we also expanded to ultramarathon runners, people that run 100 miles at a time, as well as rowers training for the Olympics. And this was really to get an assessment of the functional pathways in these bugs. And it turns out this lactic acid eating bacteria is fairly unique, in its functional capacity. Not only does it actually eat, lactic acid, but actually converts it into short chain fatty acids, acetate, propionate, and butyrate. What's kind of exciting about that is these short chain fatty acids are actually beneficial to the host, are associated with health. I think their production is indicative many times of healthy, guts and microbiomes and phenotypes and physiology. Lactic acid, typically the metabolic pathway is, it's produced in the muscles, during anaerobic exercise. It's used as an energy source. When sort of your exercise capacity outpaces your ability to use lactic acid as an energy source, it then accumulates in the blood. I think around 90% of it is filtered through the liver and the kidney, where it's converted into glucose. But here we have a bacteria that's actually promoting an alternative metabolic pathway and converting a byproduct of exercise, which I guess is, by the time it gets to the blood, mostly waste, converting it into something that's, could be an energy source, but also anti-inflammatory and essentially beneficial to the host. So this sort of intrigued us even more and made the story even more compelling. You kind of have a bug that might be able to be used as a probiotic to convert, something that's waste from exercise into something beneficial for the host. We took it a step further. Obviously, I think there's a lot of microbiome studies that are looking at correlations right now, different communities, different phenotypes, what's enriched, what's depleted. That's all cool. But I think really where the field wants to go and where it's going now is, well, what does that mean? Can you show mechanism of action? Can you show causality? So for us, we're obviously interested in not only just, observing this in athletes in a longitudinal manner, but can we actually now purify this bacteria and do functional testing? So this is just showing, after sequencing all these fecal samples, we could go back to them. We actually can culture them in petri dishes under anaerobic conditions in the lab. We're able to enrich for these bacteria. They're typically of low abundance, less than 1% in the gut, although they could still be beneficial in such a low abundance level, but we're actually able to find conditions that we could then purify them and isolate them. And we isolate several species of this particular genus, and that enabled us to do more sort of like functional validation studies. By the way, I'm almost tempted to stop at some point and ask if there are any questions in the audience, but I realize that this is all being done over, video conference and stuff like that. So I guess, that's not an option. Hopefully, I'll be able to address any questions in a follow-up. But, as I mentioned, we purified numerous strains of this genus that we discovered in, marathon runners, ultramarathon runners, and then we culture them in vitro. And, for us, the first thing we wanted to do was confirm that this bacteria could actually eat lactic acid and actually convert it into short chain fatty acids. So we grew these in sort of 96 well, deep well plates. We grew them in media that was enriched in sort of lactic acid. And then we grew them for around 48 hours. And then we actually took sort of the cultured supernatants of these bacteria. And then we could measure sort of the molarity or the concentration of lactate present in these cultured supernatants. The other thing that's cool to mention about this is, lactate threshold is actually something that's used by a lot of elite endurance athletes. A lot of times, they'll use it to sort of fine tune their training and their performance. And actually they'll prick their finger, they'll prick their ear, and they'll use sort of a monitor that could rapidly detect, how much lactate is in their blood. So if you see on the right where I have the gloves, we're actually literally using the same, monitor that athletes use in their blood. We were just using it to detect lactate concentrations in cultured media. And, the cool thing is, I think physiologically, the molarity, the millimolar concentration of lactate in the blood at resting levels is around 2 millimoles per liter. I think when you reach sort of fatigue states, it could go up to anywhere from 4 to 6 to 8 millimoles per liter. I should say this media had 22 millimoles per liter, so it was way above physiologically relevant levels. But what you could see here is that basically in culturing our bacteria that we isolated from these ultramarathon runners, within 48 hours, it pretty much wipes out all of the lactate from the system or the lactic acid. I think I'm using lactate and lactic acid interchangeably. I don't actually think they're the same thing, but I guess maybe for purposes of this discussion, I'll sort of interchange them. More than that, we then took sort of our cultured supernatants and we ran them through mass spectrometry to look at sort of short chain fatty acid production. And what we found is that actually 40 hours after culturing these bacteria in the cultured media, not only does it deplete lactate, but it then sort of actually converts it into propionate or short chain fatty acids. So this was really cool. We basically observed this correlation, in athletes and the increase of this bacteria. We purified it. We then grew it in the lab, and we found that it's able to, convert lactate into short chain fatty acids. We then took it a step further, right? So this was kind of exciting. We then want to say, okay, great. We know this bacteria could do these cool things, but what does that mean from a physiological perspective? What if we actually did preclinical testing and we sort of gave this bacteria to mice and see if it improved their performance and recovery? So this is actually a figure from the paper whereby we basically did a crossover study where we fed mice our lactic acid eating bacteria. We compared it to a control bacteria. And then basically we ran them on a treadmill till exhaustion to see if there was a difference in endurance. We did this sort of endurance testing 3 days in a row, and then we switched the groups in terms of what they were fed. And what you could basically see on the left and then on the right is that basically at every time point in which mice were fed this lactic acid eating bacteria, they showed around a 13% improvement in run till exhaustion, which was very exciting. And it's also pretty huge. I mean, I think if you talk to any athlete out there and you say, hey, we're going to improve your run time by 13%, they'd say, where do I sign up? Another thing that I want to point out from a scientific perspective is these were quote unquote normal mice. I think a lot of times in the microbiome space or area or field of research, you see people doing studies on germ-free mice. These are mice devoid of native bacterial communities, or you see them do studies on, gene knockout mice to sort of induce a certain disease state or phenotype. These were mice with their own native microbial communities that were basically fed this bacteria. So it was, I guess as close, at least in mice, as you could get to a real physiological setting. So I think the fact that we're able to observe this functional effect was pretty exciting to us. So this was, obviously great for us. The other thing to point out too is, we collected blood from these mice 24 hours after exercise, and then we looked for inflammatory cytokines. And basically what we observed is almost across the board, there was a significant reduction in these cytokines that are associated, with inflammation, TNF alpha, interferon gamma, interleukin 6. So again, this was exciting for us because we basically started off by working with athletes, sequencing their microbiome before and after the marathon, observing an increase in a bacteria that eats lactic acid. We then purified this bacteria. We confirmed in vitro it eats lactate and then converts it to short chain fatty acids. We then fed this bacteria to mice and observed a 13% improvement in endurance and then a reduction in inflammation after exercise. So this was pretty mind-blowing to us, and it was very exciting. And I think, if you look at FitBiomics and what we're trying to do now is we've made these cool scientific discoveries. Our goal is to try and translate this now into real-world applications. Imagine having sort of a consumer-facing probiotic that could be used not just by elite athletes, but everyone that maybe wants to be more active or increase exercise capacity, or even applications beyond athletics. So I think, this was really cool, and it validated the notion of, mining elite physiology to look for next generation probiotics. I kind of mentioned earlier, I grew up in the comic book industry. I'm sort of a geek and I'm a huge fan of Marvel, but so, it's cool to have a little bit of fun with this. I think, most people now know Captain America, and the notion of sort of having these cool superpowers, and a lot of that was, developed because of taking a super soldier serum that basically gave Captain America all these crazy powers as far as strength, endurance, and recovery. And what's kind of cool is, we're working with elite athletes, and if you just look at like Wikipedia or Google and Captain America and their background, and just says here, he has the strength, the speed, the endurance, and reaction time superior to any Olympic athletes. And then if you actually go a step further, most notably, his body eliminates the excessive buildup of fatigue-producing poisons in his muscles, granting him phenomenal endurance. And then it goes a little bit into the superserosurgeon. If you actually click on fatigue-producing poisons, what comes up? Lactic acid. So I like to joke with people, and this is mostly because of my background in pop culture and, comics and stuff like that. Maybe what we've done and what FitBiomics has done is we might have actually discovered perhaps one of the ingredients for a super soldier serum. So maybe we could help make, the next Captain America, or at least just make super fit individuals. So, for me personally, I think that was an exciting accomplishment, if nothing else. Speaking of pop culture and, community and people being excited about what we're doing, I think the excitement that this paper has generated has sort of been even beyond our, I don't know if I want to say wildest expectations, but beyond our expectations. I think this, might have been the highest covered, publication coming out of George's lab and the Wyss Institute, which is kind of saying something because George Church gets a lot of attention. But this was covered by over 130 media outlets across the world, New York Times, LA Times, Discover, CNN, NBC. I think it speaks to, again, this notion of, yes, we're doing really cool cutting edge technology, but, sort of using athletics as a medium to make it more relatable to the general population. And I think, again, what are the applications? It extends beyond therapeutics or disease, but for just general health, wellness, and fitness. So I think what's really exciting for us, and again, coming from my background in athletics and sort of pop culture, it's, again, translating this science into real world applications that everyone can sort of understand, be inspired by, but also have access to. I'll also just say, a few more things. This is sort of extending beyond, I guess, the publication now. Obviously FitBiomics is, a startup, and we're looking to sort of develop consumer-facing products. I think there's a huge opportunity. And this is really sort of the transition going from academic research to now sort of startup phase and industry, developing technology, not just for the sake of it, but developing technology that has real world applications and that could be useful to people. Like we talk about microbiome, we talk about beneficial bacteria, but probiotics, it's a huge market, a huge industry. It's a $60 billion industry, which is kind of amazing. And you think about sort of, how this could be disrupted with new innovations and new technologies. Also, what's kind of cool here is, 90% of this industry consists of two types of bacteria, lactobacillus and bifidobacterium. I think if you contrast that with the trillions of bacteria in and on our body that help promote our health and development, there's a huge opportunity to use technology to discover next generation probiotics that can sort of improve and disrupt this industry. Certainly the lactic acid eating bacteria is one of them, but maybe even within these sort of bacterial groups, there are ways for improvement. And, I'll just transition sort of briefly some of the other things we're doing. I mentioned lactobacillus and bifidobacterium. These are some of the common species and strains that are in the market. Here's kind of something that's interesting and cool. We're actually really good also at isolating these sorts of probiotic species from athletes, right? So again, we could sequence their biomes. We could look for lactic acid eating bacteria. We could look for other things that are unique and enriched. But also, this might be a good place to start with these bacteria because they're widely used in the industry and the market. They have a lot of sort of, value from a commercial as well as even a health, digestion and immunological perspective. So, we could isolate these bacteria and, some of the things that we're discovering is that athletes have their own unique genetic variations of these common existing probiotic strains and species. The way I like to say this is, if you isolate a lactobacillus from a Chobani and compare it to one that's isolated from like a Serena Williams, they're going to be vastly different probiotics and microorganisms. And I think this makes sense because these bacteria naturally evolve and cater to the physiological demands of their host. I think one thing that we're excited about is, again, everything we're doing is understanding optimal health, optimal physiology, optimal performance, and then sort of isolating next generation probiotics that are sort of associated with that, help drive that, help support that. So, even from this very basic level, right, if it's a $60 billion probiotic market that consists of mainly lactobacillus and bifidobacterium, okay, great. How can we mine the diversity of nature and biology to look for, improvements over the status quo? So I think there's a lot of cool things that FitBiomics are doing, from just, probiotics for general gut health, probiotics for endurance and recovery, and then continuing to expand our platform to even look for, what's next out there, because I think we're just scratching the surface. And, this is kind of a joke, but, ultimately for us, the goal is, it's great. We could discover these bugs, we could purify these bugs, we could functionally test them, but ultimately we want to turn them into food, beverages, and supplements. Think of a kombucha, think of a yogurt, think of a pill or a supplement, and basically, can we sort of use a diversity of these delivery modalities to turn these next generation probiotics derived from the most fit and healthy people in the world into consumer products for the masses to sort of have access to? There's a lot of, I skipped something, I think. There's a lot of applications with what we're doing. I mean, even you think about from an athletics perspective, just having access to this data, right? I think when we work with athletes, they're excited, about the technology. They're excited about learning more about their biology. They're excited about seeing how it changes over time. They're excited about how this might be used to optimize their training, their recovery, their performance, how it can extend their careers, and what applications it has for maybe personalized nutrition. Now, one thing I do caution and say is that, we're at the very beginning, of this field, and, this new frontier. So I think there's a lot that we still have to learn. And I think for us, it's important not to overpromise. I think it's important to engage athletes and engage the community and, be candid and honest about the technology and where it is. But we also have to start somewhere. I think the more data we have, the more samples we have, the more athletes we work with, the more we're going to learn. So I think this Nature Medicine paper is a first example of all the cool and exciting things that, we could discover. And then obviously from a commercial perspective, turning those into probiotics, turning those into like ingredients for food and beverage modalities. So that's exciting. We're in the real world now, and I think we're trying to translate discoveries that we made at Harvard, but also what we're really excited about, and being a former athlete myself, is continuing to engage, in the athletic community. And that's working with, Olympic champions, national level champions, professional athletes, and really building this community. People that are excited about the science, people that are excited about sort of translating this into, products that could promote health and fitness to the masses. And this is, again, these are collaborations between the athletics and science communities that we're looking to foster and grow and develop. So I'll sort of transition from there. Obviously, you need a team to achieve the dream. And I think for FitBiomics' perspective, what we're excited about is the diversity of our team. And it's an eclectic group of, scientists, people in athletics, people in business, people in marketing, people that could help us tell our story. Because I think, as we spoke about earlier, science is great, but you need to find a way to communicate that and convey that to general audiences. You see George Church here, who was my mentor at Harvard. You see Alex Kostik here, who was my collaborator when, he's a professor at the Joslin Institute. But, we're really excited about what we're doing. And, I like to show this. So is George. This was one of my last days at Harvard when we were getting ready to leave and spinning out FitBiomics. I brought a basketball in, and I think George was exciting. It was actually raining that day, and George wanted to go out and play basketball. And I was like, I think we'll pass on that. But, I think we're excited because, again, I think we're used to doing a lot of, George especially, developing cutting edge technology that could be helpful to people. But I think, what we really need is to find a medium to actually translate that to the masses, to make it popular, to educate people, to help people. And it doesn't always have to be therapeutic or disease-based. It could be general health, it could be wellness, it could be fitness. I always say George Church, is one of the founders of modern day genomics, and he has around 20,000 followers on Twitter. Someone like LeBron James has 40 million followers on Twitter. So I think there are definitely mutually beneficial collaborations that could be developed between the athletics and science communities. I'll just kind of finish there. As mentioned, we're, an early stage startup company. We're based in New York. And we're excited, right now with the technology that we're developing, translating discoveries that we just went through into real-world applications, continuing to build our community, continue to work with athletes, and all sorts of folks. So I think for anyone out there that's interested in learning more, please get in touch with us. You could sort of scan this code while it's up on your screen. It'll direct you to our website. There's a lot more information there. You could reach out to us via email. You could also follow us on social media. And I think, we're really excited about, building the community, the athletic community, people that are interested in the products or the probiotics, people participating in the science. So, we're excited. We appreciate the opportunity to present this research to you, and we definitely look forward to, talking with many of you and engaging and continuing discussion in the future. So I'll kind of stop there. I spoke a lot, but, thank you for listening, for those of you that made it to the end, and thank you for the opportunity, and looking forward to having a chance to speak with many of you down the road. Thank you.
Thank you, Dr. Scheiman, for that informative presentation. I would also like to thank Labroots for making today's educational webcast possible. As a final reminder, Dr. Scheiman will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.